Hello, everyone. It's a great pleasure to share with you a little bit about my work on brain stimulation and motion sickness. Brain stimulation refers to delivering physical energy from an external device to the brain to alter its state. In this study, electric current is the form of physical energy used. Electric current brain stimulation has been extensively studied in human participants for 6 decades and is considered a gold standard for investigating causality in neuroscience. This is because it forms a closed-loop framework for studying the brain. Simply put, one half of the loop involves brain reading through EEG, while the other half involves brain writing through brain stimulation. Brain stimulation is a promising approach for mitigating motion sickness. Unlike other motion sickness mitigation techniques that rely on modulating peripheral physiological states, brain stimulation treats motion sickness as a brain state problem. All parameters used in brain stimulation are based on causal links with EEG biomarkers. For example, if an EEG biomarker shows an increasing trend associated with motion sickness, brain stimulation can reduce symptoms by delivering electric current to interrupt that trend. Most importantly, brain stimulation aims to enhance a person's resistance to motion sickness without requiring any redesign of existing VR content. To put it simply, brain stimulation can be thought of as an electric version of the traditional motion sickness pills. There is a growing number of brain stimulation-based motion sickness mitigation studies. Although many of these studies were motivated by the idea of vestibular inhibition, and authors reported significant mitigation effects. However, none have provided direct evidence that the vestibular system was actually inhibited. This study aimed to address that gap. The hypothesis is that if the vestibular system were truly inhibited by brain stimulation, then involuntary gaze instability would be observed in the brain stimulation group, but not in the sham brain stimulation group, because the vestibular system is well known for its role in gaze stabilization. As for why vestibular inhibition might reduce motion sickness, because motion sickness is thought to arise from the conflict between the visual and the vestibular systems, according to the multisensory reweighting theory. So inhibiting the vestibular system effectively reduces the relative weighting of both visual and vestibular cues, thereby lowering the conflict that causes motion sickness. So this video shows our experimental setup. You can see that the participant was wearing a VR headset and performing a tunnel travel task. At the same time, the participant's body was passively rotated in a rotating chair. To reduce motion sickness, the movement profiles of the tunnel travel task and the rotating chair were deliberately designed to be mismatched. You can also see that participant was wearing a cap with brain stimulation electrodes attached. This is the detailed experimental procedure. It consists of 4 blocks, 1 practice block, 2 resting state blocks before and after 1 brain stimulation block. The stimulation block could be either real or sham. Within the brain stimulation block, there are 6 rounds. This means participants completed 6 3-minute continuous tunnel travel tasks with a 2-minute break after each round. The gaze data was collected in real time during each round using the VR headset's built-in eye tracking device, sampled at 100 20 Hz. We follow open science practices. The eye tracking data from this study can be downloaded for free from the link provided. The date format looks like this. Most of the column titles are self-explanatory. Among them, the values highlighted in red are the variables used in this study. These include the gaze vector in the X, Y, Z directions, the left and right eye openness indicators, as well as their corresponding confidence levels. We consider a gaze vector to be valid only when both the eye openness indicator and the confidence level are equal to 1. There are two outcome measures in this study used to quantify gaze instability, BCEA and SPVA. BCEA is a spatial metric that quantifies how widely the gaze vectors are spatially scattered. SPVA is a temporal metric that quantifies how quickly the gaze vectors drift over time. As mentioned earlier, we compare these outcome measures using a between group design, that is active group versus the sham brain stimulation group. Regarding the results, I would like to begin with this figure. It presents the distribution of side effects induced by electric brain stimulation, including tingling, itching, and burning sensations. And as we can see, the red dots representing the average scores for the 3 types of side effects fall between 0 to 2 on a scale of 0 to 10. I'm showing this first to highlight the feasibility and the safety of brain stimulation for real-world applications. In other words, we do not need to be afraid of using brain stimulation. This figure shows the BCEA results from 2 participants in the active and sham brain stimulation groups, respectively. As we can see that the participant receiving active brain stimulation displayed greater gaze instability across all runs from run 1 through run 6. This figure shows that the SPV results from 2 participants in active and sham brain stimulation groups, respectively. Similar to the BCEA findings, the participant receiving active brain stimulation displayed greater gaze instability across all runs from run 1 to run 6. And this figure shows the group level grand average results. We can see that SPVA showed significantly greater gaze instability in the active brain stimulation group at rounds 1, 3, and 4, while BCEA showed significantly greater gaze instability in the active group at rounds 2, 4, and 6. Because the self-reported motion sickness scores, that is FMS, showed a significant effect at rounds and 4, we can say that BCEA, the spatial metric, had a more delayed or lingering response. However, SPVA, the temporal metric, indeed showed a more rapid response than BCEA. So the significance of this study is that we demonstrated how gaze instability can serve as a simple and objective marker for brain stimulation-induced vestibular inhibition, building on the vestibular system's well-established role in gaze stabilization. This provides an intuitive and quantifiable explanation for how brain stimulation can mitigate motion sickness in VR. We also can discuss whether this approach may be more practical or feasible than the newly proposed EEG-based neurofeedback method reported in Advanced Science last month. Thanks to the team and our founders for their support, and thank you all for listening.